Good morning to one and all. I am Vadarevu Soni from Department of Home Science. I'm going to deal with the subject health, hygiene and wellness. In that I'm going to cover the second unit. It is a continuation of it. So we already spoke about the bacteria definition, its shape and size. In this we're going to deal about the structure, nutrition, respiration, growth and reproduction. Coming to the structure, if you see, as already we spoke about the structure, we spoke there is no nucleus at all for the bacteria. So in the cell, bacteria cell, we have two layers. One is outer layer and one is inner layer. The outer layer is also called as cell envelope, and the internal layer, which has all the plasmids, nucleoids, ribosomes, or DNA or cell wall, whatever it is. The outer wall is called as cell wall. So let us talk about the cell envelope or the cell wall. So the main structure of the cell already we discussed gives the structure. Because of this it gives a structure. The cell envelopes includes the cell wall of the bacteria and the plasma membrane beneath it. So the outer envelope acts as a structural and physiological barrier protecting the interiors of the bacterial cell from the outer external environment. The function of the cell envelope is to protect the bacteria from the osmotic lysis and gives the bacteria its shape. Right. What are the cell interiors? The internal structure of the bacterial cell consists of the protoplasm, which consists of cytoplasm, cytoplasmic inclusions, mesosomes, ribosomes, inclusion granules, and single circular DNA. Additional, they have capsules, they have flagella, fimbria, and spores. So let's go into the depth of the structure. So this is a clear picture of a structure of a bacteria cell. So you can see there is a cytoplasm, there is a plasma membrane, there is a cell wall, there is a flagellum, there is a plasmid DNA, bacterial DNA, and ribosomes, inclusion membranes, and mesosomes. So let us go individually. So what is cell wall? The cell wall of the bacteria are made up of glycoprotein. It is a protein in nature. The main function of the cell wall is to help in providing support, mechanical strength, and rigidity to the cell. It also helps in it also protects the cell from bursting in a hypotonic medium. Now coming to the plasma membrane. It is also known as cytoplasmic membrane or cell membrane. It is composed of phospholipids, proteins, carbohydrates forming a fluid mosaic. It helps in the transportation of substance including the removal of waste from the body. It helps in providing a mechanical barrier to the cells. Plasma membrane act as a semi-permeable membrane allowing only selected material to move inside and outside the cell. What is cytoplasm? It mainly helps in cellular growth, metabolism, and replication. Cytoplasm is a storehouse of all the chemicals and components used to sustain bacterium life. The ribosomes, if you see the picture, you have 80 ribosomes, 50 small S units, that is large subunits, and 30 small subunits. See, they are tiny granules made up of RNA and proteins. They are the site of protein synthesis. They are free floating structure that helps in transferring the genetical code. Mainly it is used for genetical transformation. Then what are plasmids? Plasmids are a small circulars of DNA. Bacterial cell have many plasmids. Plasmids are used to exchange a DNA between bacterial cells. It is mainly for the exchange purpose of the DNA from the bacterial cell to the plasmids. Now come to the flagella. It is a rigid rotating tail. As I already said, it is a tail. If you see the tails, there are different types of tails. It helps the cell move clockwise and anti-clockwise forward and also helps the cell to spin. The rotation is powered by an H positive gradient across the cell membrane. Mainly the flagella is mainly used for locomotion, which is also called as movement. From one place to other place, the flagella is mainly used for it. The pili. The pili is nothing but it is very compared to flagella, it is short in shape. If you see the brown color in the next picture, it is a flagella. It is smaller than the flagella, fixes bacteria to the surfaces. It is mainly getting attached onto the walls of any kind of a surfaces. It also helps in reproduction during conjugation. So it is mainly called as reproductive villi. Then what is capsule? If you can you see the difference between a capsule and a slim layer? Yeah. Capsule is a kind of a slime layer which covers the outside of the cell wall. They are composed of thick polysaccharides. Polysaccharides are nothing but carbohydrates classification. It is used to stick cell together and work as a food reservoir. It protects a cell from dryness and from chemicals. Right? So this is the structure of a bacteria cell. Now, nutrients sources in bacteria. As a bacteria is also as like a human being requires all the kind of nutrients. They require both macronutrients and micronutrients. The macronutrients are, you can say it as carbohydrate, protein and fat, and the micronutrients are like you can say nitrogen, hydrogen, oxygen, phosphorus, sulfur, iron, all these things. The bacteria also grows along with the food or the nutrient intake. So let us see what it is. Bacteria necessarily require macronutrients such as carbon, nitrogen, hydrogen, oxygen, phosphorus and sulfur to build important biomolecules, that is protein and nucleic acid, that constitutes most cellular component. So let us see the individual role of different micronutrients. So carbon is mainly essential micronutrients that constitute a significant portion of the cell. It builds biomolecules like carbohydrates, protein, nucleic acids and lipids. Nitrogen is necessary to build protein and nucleic acid. Bacteria may utilize atmospheric nitrogen or other inorganic forms. Hydrogen and oxygen constitute many organic compounds and they are part of water. Phosphorus is necessary to synthesize nucleotides and phospholipids. They are nothing but protein in nature. Sulfur is a part of amino acids, examples like cysteine and methionines. These are the examples of amino acid, and amino acid is a combination of a protein. Besides all the above, potassium, magnesium, calcium, sodium are essential to maintain the bacterial cell structure and functions. Bacteria need micronutrients like iron, boron, chromium, magnesium in traces or minute amount to perform electron transport chain reaction and other processes. Total breakdown of glucose, and in anaerobic respiration partial breakdown of glucose is being formed, and finally the end product of aerobic respiration is carbon dioxide and water, and for anaerobic it is carbon dioxide and lactic acid or alcohol. Fermentation takes place in anaerobic reactions. So if you see, that is a difference between aerobic and anaerobic respiration. So let us see what how do you define it. So in aerobic respiration, it is a type of a cellular respiration that takes place in the presence of oxygen to produce energy. It is a continuous process that takes place within the cell of an animal and plant. The process can be explained with a chemical formula. If you see, glucose plus oxygen gives rise to carbon dioxide plus water and energy. Like anaerobic respiration, it is a type of cellular respiration that takes place in the absence of oxygen to produce energy. The glucose gives rise to alcohol plus carbon dioxide and energy. Right. Now let us see the main important phase. It is a growth phase. Okay. There are four phases in which where you can say a lag phase, log phase, stationary phase and death phase. So if you see a picture where the graph, the first phase is lag phase. If you see a straight line, okay, if a straight line is being seen, this is a lag phase where there is no increase in number of living bacteria cells. This is a growth of a bacteria which I am talking about it. In the growth of a bacteria, if you see, first phase, lag phase, where there is no growth, means there is no increase in number of living bacteria cell, that is called as a lag phase. If you see the second phase, it is called as a log phase, where there is a growth. Can you see the growth over here? There is a slanting line seen. It increases in the number of living bacteria cell, that is called as log phase. Then comes to a stationary phase, that is a plateau. Can you see a plateau of here? A plateau in number of living bacteria cells, rate of cells division and death roughly occurs. So here if you see, there is no growth happening in the stationary phase. After certain period, slowly the bacteria cell starts to decline or death may happen. That is, this is a decreasing line you can see. There is a decrease in number of living bacteria cells, and finally the bacteria cell gets died. So this is the main difference between the growth phase of bacteria growth curve, right? Next come to the temperature. As we know, different types of bacteria live in different temperature. They can live at any temperature depending upon their texture. So let us see. Again, they are classified into thermophilic, thermoduric, mesophilic, psychrophilic, and cycloduric. If you see the picture over here, what is psychrophiles? They are very cold loving. They can stay, they can withstand to a cold temperature rather than a heat temperature. So they can withstand to a temperature to 10 to 20 degrees temperature. They are called as psychrophiles. Then what are mesophils? That is a moderate temperature. Loving optimum temperature should be 25 to 40 degrees temperature. You can think our temperature where we are, Visakhapatnam temperature. Right? The thermophils, it is a heat loving way they can withstand to high temperatures like 50 to 80 degrees temperature. And hyperthermophiles means extreme heat. They are called as minus 80 degrees or more than that. It is 80 degrees temperature. They are called as uh, hypothermophiles, right? So these are being classified into different parts. That is about the temperature of growth microorganisms, right? Then bacteria. Yes, bacteria can reproduce. Yes, bacteria can reproduce into two ways. One is asexual reproduction by a binary fission, and sexual reproduction is in a bacteria. Let's see about it individually. Asexual reproduction occurs by binary fission, where in a cell divides into two and the formation of endospore occurs during the unfavorable conditions. So let us see deep into that. So this is a asexual reproduction which is formed by a binary fission. So see, if you see the picture, it's very clear. A single cell, when it comes to asexual reproduction, the DNA which is there, it starts replicating. Slowly the cell wall and the plasma membrane begins to divide, right? Then a septum is being formed between the single cell, because there are two DNA, equal shared DNA, and the cell wall and the plasma membrane is being divided, and complete section cross section is being done, and slowly a individual bacteria cell is completely divided into two equal DNAs, right? This is how you can see. Now they both separated into an individual cell, right? So it is a common and simple method of asexual reproduction. In this process, asexual reproduction, the parental cell divides into two, leading to equal sized daughter cells. Under favorable condition, bacteria multiplies rapidly and may double into 10 to 15 times. So this is a simple method of asexual reproduction. Right. Now coming to the sexual reproduction. Again,